Hello, welcome to this tutorial on charge measuring electronics in medical applications. The measurement of electrical charge is involved in a broad variety of applications. For instance, charge can be exchanged at an electrochemical interface between a solid electrode and an electrolyte, an ionic solution containing molecules, for instance, through redox reactions. Charge can be put in motion during an impedance measurement. And finally, charge can be generated by the absorption of a photon in a semiconductor, generating an electron-hole pair. In this tutorial, we focus on the application in the biomedical field, in particular at the microscale. This is motivated by a convergence between the capability of microfabrication, enabling deep submicron lithography of artificial devices that can match in size with single biological entities, such as biological cells, macromolecules, DNA molecules, proteins, and so on. This convergence has a common goal of developing portable, miniaturized diagnostic platforms for point-of-care lab-on-chip diagnostics. Here we will show some examples of application of current sensing, the measurement of current versus time, with current typically in the picoamp range with a bandwidth in the order of kilohertz. Of course, the collected charge, the current, depends on the electrode area and the concentration of the different molecules. Those molecules can be neurotransmitters released through the mechanism of exocytosis from neurons, cells, and electroactive. Even if a molecule is not directly detectable through a redox, in some cases it can still be detected by means of amperometry using some mediation. In particular, this is the case of glucose, which is of course very relevant for diabetes, that can be bound to an enzyme, and one of the byproducts of the catalysis of the enzymes, glucose oxidase, is hydrogen peroxide that can be detected electrochemically. Other examples include a patch clamp, a technique to directly measure the current flowing through an individual ion channel, a transmembrane protein present in neuron cells, or the measurement of molecules translocating through nanopores, extremely small openings separating two conductive chambers filled with conductive liquid, for instance for automatic sequencing of the DNA strand. There is a broad family of affinity biosensors, such as DNA, immunosensor with antibody antigens, and so on. In all these cases, the binding of the analyte with the probe can be detected by a change in the impedance of the electrochemical interface. At the micrometer scale, the growth of a colony of cells sitting on a planar electrode can be monitored by means of impedance, and also in a dynamic case, the flow of cells in a microchannel can be detected by means of impedance flow cytometry. So far we have discussed examples of diagnostics based on the extraction of molecules from samples, from physiological samples such as blood, urine, saliva, and so on. Of course, in some cases, those molecules that we want to identify are within the body in specific organs. And in this case, we need different diagnostic tools, which fall in the family of medical imaging techniques. Of course, there are many techniques like impedance tomography, infrared spectroscopy, echography using sound waves, CT using X-ray that penetrate through the body, magnetic resonance, and nuclear imaging, in particular scintigraphy, that use gamma ray to probe the presence of such molecules inside the body. In this case, the probe is chemically attached to a radiotracer that is a small molecule that has a radioactive decay and emits gamma rays. From outside of the body, by using a gamma camera, that is a camera imaging at the high energy of gamma rays, we can take images around the body, around the patient, and by tomography reconstruct the exact position of emission of the gamma ray, which correspond to the accumulation point of the pathology that is molecularly identified by the molecular probe. There are two mainly techniques: PET, positron emission tomography, and SPECT, single photon emission computed tomography. In medical imaging, there is an emerging trend that is the combination of multiple imaging techniques that is called multi-modal imaging. In particular, it's extremely relevant to combine techniques that provide anatomical details, such as CT and MRI, with techniques that are molecularly specific, like SPECT and PET. The idea is to measure at the same time the position, the exact location of the pathology inside the body, saving time for the patient and also offering better quality to the doctor. Here, the role of electronics has to face different challenges. One of the challenges is, of course, the compatibility of electronics, for instance, in the case of the gamma camera, with the magnetic field of a magnetic resonance scanner. Here you can see one prototype. This is a clinical system that has been developed. It's a SPECT insert for neuro-oncology that has been developed recently in a European project called INSERT. And here the challenge was to build electronics compatible with the strong magnetic field and to grant mutual compatibility between electronics in the SPECT scanner with the MRI, with the standard commercial MRI scanner. And this had impact on the choice of materials to avoid ferromagnetic components and also correcting the distortion in the magnetic field due to the collimator for SPECT gamma rays, while on the SPECT side electronics, particular filtering techniques and spatial layout techniques were needed to avoid disturbances induced by gradients and electromagnetic interferences. Now let's focus on the metrological aspects of this instrument. Typically, these current signals come in form of pulses or steps, and we want to identify the amplitude of the area of these pulses, which corresponds to the charge of these events. The two main metrological properties are accuracy and resolution. Accuracy is typically addressed by means of calibration, which can be linear, simply adjusting gain and offset, or nonlinear, using typically higher order polynomial or lookup tables. It is important to keep under control cross-sensitivity to temperature or other effects, and also, in the case of multi-channel system, to control crosstalks among parallel channels, especially in high-density implementation. Resolution is the capability to discriminate the smallest variation of the input signal and is determined by noise. Here, with noise, we consider the intrinsic electronic noise of the components inside the instrument, whose overall effect depends on the circuit topology and also on the bandwidth. For other sources of fluctuations like external interferences, of course, we can use shielding and filtering to reduce their impact in the system. This is the typical acquisition chain. We have an analog front end, which converts the input current into a voltage, then a filter that maximizes the signal-to-noise ratio, and then an analog to digital converter, which digitizes the signal, which is typically then processed by a digital platform that can be a microcontroller or an FPGA. The most relevant figure of merit in the design of this chain is the signal-to-noise ratio. Of course, we would like to maximize the signal, and this can be done in some cases, for instance, acting on the detection technique, as in the case of readout cycling. And of course, we want to minimize the noise. And this can be done acting both at device level and circuit level, as we will see here. The most common type of front end is a negative feedback amplifier, the transimpedance amplifier, that provides several advantages: the linearity in the conversion from charge to voltage, low input impedance thanks to the negative feedback, and neutralization of some parasitics connected to the input node. However, there are some critical aspects, such as the stability and the sensitivity to the input impedance. If we perform the noise analysis, it turns out that, of course, the feedback resistor has an impact in the noise spectrum, but one of the most dangerous terms is the total input capacitance connected to the input node, which has to be minimized. The minimization of this capacitance can be performed by reducing the electrode area, but of course this implies a decrease in the signal unless we have an active way to capture the charge. We can reduce the interconnection parasitics by shortening the connection, avoiding shielding if possible, and choosing insulating substrate instead of conductive substrates. Or in some specific cases, we can use also a reactive cancelling technique by adding an inductor, which at resonance can cancel the capacitance, but of course, this is only possible when operating at a single frequency, as in impedance tracking. The ultimate level of interconnection reduction is to have the electrodes and the electronics on the same microelectronic chip. This is a clear trend, and there are plenty of technologies that enable to fabricate on the CMOS platform several types of transducers: charge-based, photon-based, magnetic field, mechanical, micromechanical, nanoscale devices, and so on. Some open issues are materials and packaging. This is an example in the growth of the number of channels that can be fit in a single chip, from 1 to more than 2,000 in the last years. This is all for application to biomedical, to molecule detection. Then it's possible to act at circuit level by adopting some advanced topology that can overcome the trade-off limiting the transimpedance stage, for instance using the cascade of an integrator and a differentiator, or completely removing the feedback resistor and using, for instance, periodic reset to discharge the input capacitance. There is one case when the negative feedback gets in trouble, that is when we have a large charge to collect related to a large device capacitance and large bandwidth. This is the typical case of the readout of silicon photomultiplier. In this case, negative feedback stability is critical, and we have proposed the use of a weakly positive feedback to enable stability also in these cases. Interesting, impedance measurement is a special case of current sensing, where we have our unknown impedance and we add a forcing generator. In order to extract the information of impedance at a specific frequency, we can use a synchronous demodulation, a lock-in scheme, which enables the best performance in terms of signal-to-noise ratio, thanks to extremely narrow bandwidth that can be tunable at different detection frequencies. In the case of impedance, we have a couple of additional noise sources that has to be taken into account. First of all, the noise of the signal generator, which excites our unknown impedance. This will produce a current noise that gets into the chain. In order to reduce this effect, we can adopt a differential readout scheme. In particular, this is suitable in the case when we can fabricate very well matched devices, or we can use some kind of active tuning in this type of calibration. Finally, a second source of uncertainty and fluctuation limiting the performance of digital lock-in amplifiers are the 1/f slow fluctuations of the reference voltages of ADC and DAC used in this implementation of the lock-in. In order to overcome this limitation, we have shown that it's possible to use a ratiometric technique where two ADCs are operated in parallel by fast switching of the input signal going through the two paths, and then reconstructing at the end and taking the ratio so to cancel the common effect due to the slow fluctuations of these reference voltages. In conclusion, we have highlighted how current and charge measurement are ubiquitous, in particular in the application to biomedical fields, diagnostic and molecular identification. In order to reduce noise, the minimization of the input capacitance is critical, and this can be often done by miniaturizing the electronics interfacing with the electrodes down to the limit of putting them on the same chip. The transimpedance amplifier is the best circuit solution, but it's a basic one. More advanced solution can be adopted in terms of addressing the noise versus speed trade-off. And in some cases, negative feedback fails, as in the case of the silicon photomultipliers that are used, for instance, for SPECT, where we have other system-level challenges of mutual compatibility between electronics for gamma-ray imaging with the magnetic resonance interfaces. Thank you for watching.